Welcome to your second video in your Chapter 4 video lecture series, where we're going to talk about Section 4.2 on the chemical structure of the plasma membrane. And here are your learning objectives. First, we're going to talk about lipid components. Our plasma membrane has a fluid mixture composed of equal parts of lipid and protein by weight. We're going to be able to regulate movement of most of our substances in and out of the cell, and we'll see that we have a ton of different types of lipids. We have phospholipids, we have cholesterol, which is pictured in the yellow here. They're tiny structures, and we also have glycolipids. First, we'll discuss the phospholipids. This is what people know as the balloon with the two tails. You can also see it pictured here, balloon and our two tails, or it's the ball with the hairpin attached to it. We say that the head is polar and hydrophilic, meaning water-loving, and our two non-polar tails are hydrophobic, meaning they are water-fearing. They form two parallel sheets of molecules lying tail to tail. So you could see that sheet pictured here on the right, where we have one phospholipid over here, another one lined up next to it, and so forth. They're lined up like soldiers, right? And then we're going to have another row in the opposite direction so that we form this, this phospholipid bilayer. Our hydrophobic tails form this internal environment here so it can hide from the water, and our hydrophilic heads are going to be directed outward. This phospholipid bilayer is the basic structure of the framework of our plasma membrane. It helps ensure that our cytosol is remaining inside the cell, and our interstitial fluid is going to remain outside of the cell. So this is a little bit confusing over here. This is one cell pictured in the lower right corner, and this is another cell pictured in the upper left corner. So this is showing the cytosol of this cell, whereas over here we have the cytosol of another cell. And this orangish color in the middle, that is our interstitial fluid. It is our external environment. And the darker brown or purplish lines here, that's representing the plasma membrane of each cell. Now for cholesterol. Like I said, these are the yellow structures in here, the ringed molecules, and these are scattered within the phospholipid bilayer. You can notice they're closer to those tails that we had. And it helps to strengthen the membrane. It really stabilizes that membrane against any extreme temperature changes. And the other lipid component we have are glycolipids. These are lipids that are attached with carbohydrate groups, hence the glycoside is referring to the carbohydrate portion, and it's located on the outer phospholipid region only. So you can see really just this one pictured over here, and it helps to form glycocalyx. And glycocalyx has a lot of different functions depending on what area of the body we're talking about. It can protect from stress of blood flow within endothelial cells in our blood vessels. It can also aid in cell attachment that helps our immune system and even helps in blood clotting. Now let's discuss membrane proteins. I'm sure you noticed these large blue blobs in here in our plasma membrane. They are half of our plasma membrane based on weight, and they are able to kind of float in here and move in this fluid bilayer. Almost think of them as like a rubber lifesaver in water is what you want to think of. And they're going to perform most of our membrane's functions. And we have two structural types, integral and peripheral. So our integral proteins are the ones that are embedded into that phospholipid bilayer. So you could see them going across that bilayer. and their hydrophobic regions are going to interact with the hydrophobic interior. And the hydrophilic regions are exposed to the aqueous environments on either side of the membrane. A lot of our glycoproteins with the carbohydrate are going to be attached there too. And the other type of protein we have is a peripheral protein. These, you can notice, they are not embedded in the lipid bilayer. They're just loosely attached to the external or internal surfaces of the membrane. So if we wanted to identify these proteins based on their functional categories, we can call some of them transport proteins. These transport proteins are going to work to regulate the movement of a substance across the membrane. So they can serve as channels where we can see, let's say, ions moving through. They can be carriers in which we can move substances through, or they can be pumps where we need to attach and then let go of the ion or whatever it is moving in here. We can also have symporters in which we can move two substances across at the same time, or they could be antiporters in which we are going to move substances in opposite directions. Now we can also classify them as cell surface receptors. Most of these are going to bind a molecule known as a ligand. So you could see that picture down over here where you can imagine a ligand being something like a hormone attaching to a protein receptor. Or it can also be a neurotransmitter that has been released from a nerve cell that'll bind to a receptor on a muscle cell to tell it to contract. Another functional category of proteins is an identity marker. This helps to communicate to other cells that they belong to the body. So if we had other molecules here, then the body would recognize it as foreign and could mark it for destruction. Some other functional categories would be enzymes. Enzymes may be attached to either an internal or external surface of the cell and can catalyze chemical reactions. So here you could see a substrate, or you can almost think of it as like a reactant, enters the enzyme, and some type of conformational change will take place and a product will be released. We can also have these anchoring sites where we can secure cytoskeletal structures or proteins to the plasma membrane. And lastly, we can have these cell adhesion proteins that are going to perform cell-to-cell attachments.